Hello students, today we are going to learn a very important concept, that is DNA replication, which is coming under your chapter 6, molecular basis of inheritance. Under this concept we are going to explore features of replication, experimental proof of semi-conservative replication, mechanism of replication. What is replication? Replication is a process in which DNA synthesize its own exact copies. Okay, come to the features of DNA. So DNA replication, first one is it is bidirectional. Bidirectional means DNA replication occurs in both directions. Then semi-discontinuous in nature. One strand in replication, one strand is continuous and another one is discontinuous. Then elongation occurs in from 5' to 3' direction. Most important is DNA replication is semi-conservative in nature. Which is proposed by Watson and Crick. What is semi-conservative in nature? In daughter duplex, one strand is from parents and another strand was newly formed. Got it? Now come to experimental proof of semi-conservative nature of replication. The experiment was done by Matthew Meselson and Franklin Stahl in 1958. They took Escherichia coli and grew it in ammonium chloride medium, where in ammonium chloride the nitrogen was a heavy isotope of N15. Okay, after several generations, the N15 nitrogen incorporated totally or fully with Escherichia coli bacteria. It was clearly figured out by a centrifugation, that is cesium chloride density gradient centrifugation, where the heavier DNA settled down towards the bottom and the lighter DNA lies towards the surface. Got it? After this, they transfer Escherichia coli, the N15 Escherichia coli, to a medium having ammonium chloride with normal nitrogen, that is your N14. Okay, Escherichia coli takes only 20 minutes for cell division or for reproduction. In first generation 1, look at it. In generation 1, after 20 minutes, only hybrid DNA are present. that is the DNA strand, the two DNA strands, one strand have N15, red one has N15, whereas the blue one was N14. Got it? In second generation, that is after another 20 minutes, that is after 40 minutes, the ratio of N15, that is hybrid, and N14, the ratio of hybrid and N14 is 1:1. After another 20 minutes, that is after 60 minutes, the ratio of hybrid, that is N15 N14 and N14 is 1:3. Another 20 minutes, that is in generation 4. In generation 4, the hybrid N15 N14 and N14 the ratio is 1:7, which clearly indicates it's clearly showing the semi-conservative replication of DNA. This type of experiment also done by Taylor and their colleagues in 1958. They use radioactive thymidine in Vicia faba. It is a bean tree. Okay. Then come to the mechanism of DNA replication. The enzyme required for DNA replication are helicase, single-stranded binding protein, SSBP, single-stranded binding protein, topoisomerase, primase. This is DNA dependent RNA polymerase. DNA polymerase and DNA ligase. Remember, in prokaryotes there are three types of DNA polymerase. DNA polymerase 1, 2 and 3. But in eukaryotes there are 5 types of DNA polymerase that requires in mechanism of DNA replication. Okay. DNA replication is divided into 3 parts. First is initiation, then elongation, then termination. In initiation, first activation of deoxyribonucleotide occurs. This first activation of deoxyribonucleotides occurs. These deoxyribonucleotides act as substrate in replication as well as it provides energy to the process of replication. Got it? Then the enzyme helicase starts unwind DNA at a particular site, that is origin of replication. Remember, helicase starts unwinding the DNA at origin of replication by breaking the hydrogen bond between two strands. Got it? These two strands are stabilized by a special protein, that is single-stranded binding protein. Okay? Then the supercoil that occurs by the action of helicase is removed or the tension is removed by topoisomerase an enzyme topoisomerase. Got it? Opening of total DNA by one stretch cannot be occur because it requires high amount of energy, so that a small portion of DNA is open in bidirectional by the help of helicase. It creates a Y shape, which is popularly known as replication form. Got it? Then the enzyme primase synthesis a small RNA primer at 5' end in both sides. At 5' end of both sides. Okay. RNA primer is required in DNA replication because without RNA primer, DNA polymerase cannot add nucleotides during elongation. Got it? Then come to the process elongation. Formation of leading strand, and formation of lagging strand. Okay, come to the action of DNA polymerase. Remember, DNA polymerase add a nucleotide from 5' end to 3' end in the direction of 5' to 3'. Okay. In 3' to 5' template, it is continuous in nature. Okay, so that this is known as leading strand. The action of DNA polymerase is continuous, but in 5' to 3' template strand, the action of DNA polymerase is not continuous due to exposure of small stretches. So DNA fragments are built. Okay, these fragments are called Okazaki fragment. Got it? These Okazaki fragments are later joined by DNA ligase. Okay, this is the process of elongation. After elongation, we know that DNA replication is bidirectional. Hence, in both directions, DNA replication occurs and it completes. This is the termination. The termination is different in prokaryote and different in eukaryote. Remember, DNA polymerase has an activity known as proofreading activity of DNA polymerase. DNA replication is highly accurate in nature because a small changes in base pair leads to mutation. The rate of polymerization by DNA polymerase is very high speed. That is, in 1 second, 2000 base pair added in both directions. Hence, it is a chance of a mismatch. That is, one mismatched base pair occurs in 1000 base pair. Then DNA polymerase moves back, remove that mismatched base pair and added the proper one and move forward. It is the proofreading activity of DNA polymerase. Okay.